Hello, this is Kelly Eversole of the International Alliance for Phytobiomes Research, and I'm really happy today to be hosting Edith Hammer and Micaela Mafla-Endara, both from the Lund University in Sweden, for a webinar. But before we get to their presentation, I'd like to give you a little bit of information about the Phytobiomes Alliance. We are an international consortium of industry, academic, and governmental scientists, and we are our work is made possible by a number of our sponsors, so I want to thank them for their support, and they also allow us to coordinate research globally and also to put on things like this webinar. Just as a reminder, phytobiomes is the concept of the complex systems of plant-based agriculture. It is the idea that we have a biome, in this case the soil, and we want to put a plant in it for agricultural purposes. And therefore, we want to understand all of the geophysical and biological components that interact with that plant and with that particular biome. So it is a very broad, complex system, and it's an effort to embrace that complexity, both in terms of our understanding and in terms of our ability to create more sustainably produced agricultural food, feed, and fiber. And in addition to these geophysical and biological components, it's also influenced by management practices. So it depends on what a grower is doing. That determines what might be the best crop or the variety, etc., for that particular biome. Our vision is that by 2050, all farmers would have the ability to use predictive and prescriptive analytics based on these geophysical and biological conditions for determining the best combination of crops, management practices, and inputs for a specific field in a given year. And it's because we want to get there that we are very interested in any kind of technology that would allow us to have a greater understanding of the soil and the condition on the farm. So just as a reminder, we have our Phytobiomes Conference coming up in September this year, and we encourage anyone from a broad range that covers the full scope of the phytobiomes to submit an abstract. And we are looking for all disciplines, all the way from soil scientists to geneticists, to microbiome, to sensor development, etc. We really want to try to bring together the global community to start thinking in a systems approach. Our next webinar will be at the end of or middle of May, and we'll be looking at rhizosphere microbiome sampling. So you can already register for that and make sure you don't miss it. Just as a reminder, this webinar is being recorded and will be posted on the Phytobiomes Alliance YouTube channel within a couple of hours. You can subscribe to our channel to never miss one of our new webinars. And I also want you to see that there is a panel for you to submit your questions. And please use the Q&A panel for submitting questions, and you can submit them at any point during the webinar and during the presentation. Also, I would encourage you to monitor the chat panel, as you'll see messages from us, and you may actually see links to some of the papers that may be referenced. Already you can download handouts from the presentations, both mine and our presenters today. And I would encourage you to never miss one of our webinars, because we have had some really exciting speakers, and I'm very excited now at this point to turn this over to Edith Hammer and Micaela Mafla-Endara, who will be speaking to us about a window to the underground. So we look forward to hearing their talk today. So I will turn it over to you. Thank you for joining us, Edith and Micaela.
Thank you very much. It's a pleasure to be invited by you. So can you see my screen?
Yes, we're good.
Great. So yes, windows to the underground. We want to talk about the hidden half of the phytobiome, the one which is hidden below ground in the dark soil, and our approach to get little windows to study this ecosystem a little bit better. I guess many in this audience have been working with soil, either as gardening or with professional activities, and you probably all have had soils in your hand and have been handling soil. But have you ever been thinking how life has to feel, how would it feel to be a microbe living in this soil, in this very, very complex environment? Oops. Soil is really extraordinary. We probably have no other ecosystem on Earth that has so many species over a limited space. And it also contains more than double the amount of carbon compared to the vegetation and the atmosphere together. This is actually making it a very important part in the global carbon budget, and also a lot of interest lately how to maybe even be able to manage this ecosystem better, to not lose more carbon from soils than we already do, but maybe even to increase storage capacity to mitigate excessive CO2 emissions. And yes, there is a lot of carbon in the soil, which is not always very easy to understand how so much carbon accumulate. If you compare the soil ecosystem to a water ecosystem, you will see that there is, yes, way more organic molecules available, also easily degradable organic molecules. For example, reviewed by Hobby and Hobby here, looking at free amino acid concentrations, which are like several orders of magnitudes higher in soils than in water. At the same time, we have much more microbial cells present in the soil as well. So we have little food, a lot of food, represented by those red cubes here, and we have few cells and many cells. Why don't all those cells eat all this food? If we add some extra food source to the microbes, for example, in form of a glucose solution, in water ecosystems, it will take hours to days for this glucose to be respired away, while if we add a glucose solution to the soil, it will be respired really quickly within minutes to hours. Why don't all these soil microorganisms don't eat the other food, which is also to a large extent also easily available chemically? We think that a lot of this has to do with this microstructure that you have in soil. This is a virtual flight through a soil aggregate produced by John Crawford in this group, based on a micro CT scan of a soil aggregate. So this pore space that we're flying through now here, this is real. The texture is made up, but the pore space, the open parts in between of the solid parts of the soil, this is real data. And we think that this labyrinth is one of the main causes for the species richness of soil and also of the excessive accumulation of organic matter that we can find. Traditionally, the accumulation of organic matter has been explained by chemical recalcitrance of the molecules themselves. But with newer studies, we have learned more about this and realized that also complex molecules as lignin and phenolics and chitin can be easily decomposed by microbes if they just get there. And we know by now that there's other processes that may be much more important for the accumulation of organic matter, and having to do, amongst others, with organometer sorption mechanisms and the physical disconnection. And this is what we are very interested in our group. The presence of this pore space labyrinth that physically disconnects microbes from the food, or also enzymes from organic matter, and electron acceptors from being able to flow freely through the system. What we usually do when we study soil systems is exactly this. We destroy this intricate microstructure. We often take a soil, sieve it, vortex it, and then use it as a averaged black box approach. And that has, of course, led to a lot of very important insights, but we think it's important to not always do this, but for once in a while also consider this intricate microstructure that we have in soil and not destroy it. If we want to study soils in their original spatial relation, we can do this by, for example, doing micro-CT in a way as if you would, for example, get a body scan in the hospital. We can scan the soil aggregates and we can see where the solid parts are, where the void parts are, and reconstruct a pore space system there. But this is very difficult to use as a manipulative empirical system to do experiments on. Then we should turn to models, to empirical models. And there have been a lot of approaches trying to have spatial structure in microbial experiments. For example, these ceramic bars, or here people have been 3D printing the solid parts of a soil. And even Lego has been used to do root experiments here. But we were not happy with any of them to give us both at the same time the visual access and the spatial resolution that we wanted to have to study microbial ecology in soils. So we actually turned to microfluidic chips. They can look like this. This is a chip that we have been using in quite some of our experiments. It's a little soft rubber polymer with imprints of different microstructures that we design for different empirical questions to study. We think that these chips represent the world at the microscale in a much better way than if we would be using plate cultures or other models. At the microscale, organisms experience physical forces in a very different way than we do in our normal life. So their life is dominated by a lot of other things that we never think about. Surface tension becomes much more strong if you are a very small organism. And also capillary forces are getting much more important, adhesion and viscous drag. And here's a little video of bacteria, those little dots, and they're swimming around in this water capillary. And they are poking this air bubble, but they will never be able to get through because of surface tension that they won't be able to overcome. So the water bubble is a barrier for bacteria that they can't overcome. We built those chips by first starting to draw spatial structures that we want to use in our experiments that we design in AutoCAD. That's a program that is also used at the macro scale for example, urban planning, and we just scale it down to the micrometer scale. We can construct, the sky is the limit, different structures that we want to test. And of this AutoCAD drawing, we produce a photo mask that we use to illuminate a photoresist spun on a glass plate, for example. And this photoresist only hardens where the UV has been in contact with the resist, and then we get a structure which becomes kind of a stamp. So we have a stamp, a mask they're called, and then we have a two-component PDMS mixture that we can pour over this master and harden it to become this rubber material. We can cut out the structures we want and then activate them in oxygen plasma to covalently bond them on a glass plate. And then we have a little chip like this that we can fill with liquids or not, and we can inoculate with soil organisms. For example, here, hyphae and the nematode. And this is how we look in the clean room when we produce the masters in security gear to not get any little tiny piece of dust onto our structures that could disturb. We think that the soil chips can be useful for quite many research-fronted questions in soil ecology, microbial ecology. We can, with help of them, simulate physical heterogeneity. We can build labyrinths and simulate different porosities of a soil. We can create chemical gradients or patches as well. And we can inoculate the chips with different microbes to study interactions, for example, in different spatial settings. Not only microbes, but even plantlets can be inserted into the chips, where we then can have the roots growing into channels and study rhizosphere interactions, for example, fungal pathogens or symbionts getting in contact with the first roots of a plantlet. And then another very interesting chip, which is not produced in our group, but by the group of Nichols, that has been able to culture way more microbes than usually possible in lab settings by having very many small microhabitats connected over a gel that allows the exchange of metabolites. So we have a restriction for dominant organisms to overgrow an area completely, reserving space also for more slow-growing species, but still letting all these metabolic interactions happen when bacteria eat the metabolic end products of other bacteria, so that we are able, those Nichols group is able to grow about 50% of the microbes that can be found in the soil, in comparison to what usually otherwise on plate cultures is possible, like a 1–2% of the microbes we can find. So that's very amazing, I think. We're not the first and definitely not the only people using microfluidic chips in soil ecology. Here's an example, for example, looking at nematode movement in different simulated porosities. And another study by Deng and al. Looking at the effect of microbial EPS on soil moisture retention, showing that E. coli EPS keeps moisture in a pore space much longer than non-EPS producing mutants. And there's also a lot of people for now studying fungi and chips. Here's a list of references that you can check if you're interested in this. Yes, our group is very interested in this phenomenon of physical soil carbon stabilization. And one question which is like a red thread through work that we're doing is, can organic matter hide in the pore space of the soil? And if so, how? The main current and recent projects that are ongoing are listed here. Christine has been working with fungal foraging behavior at the microscale. Carlos has been working with the influence of spatial structures on microbial activity and organic matter processes. Milda, a physicist who is doing microspectroscopy on soil samples. Micaela, who is here, is working a lot with in situ soil microbial interactions. And Joe, an engineer, is creating chips, including chemical patchiness lately. The first thing that we felt we needed to know more about when starting this larger project is, what is spatial availability for microbes to start with? And so we set up an experiment testing how fungi interact with space and what spatial structures they can grow through well and which spatial structures they have more difficulties with. This has been performed by Christine, and she designed this chip, which she called the obstacle chip, which is actually 5 experiments in one. Here's the inoculation area. Can you see my mouse? Yes, great. And the fungal hyphae can grow into these 5 different experimental fields, and they differ in their spatial structure. Here we have different porosities. Here we have straight channels of different width. Here we have angled channels of different angles for the hyphae to grow through. Here we have a simulation of a pore opening up. And here we had a complex, more fun obstacle course included as well. And we have been testing 7 different fungal species. And to make you orientate this a little bit better, we try to give them nicknames. We have Coprinellus angulatus, who is growing with very, very many hyphae, thin hyphae at the same time, which really reminded me of marathon runners. Then we have this dude species, psilocybe, which is closely related to some psychedelic species, which is also looking a little bit giggly when it grows. I will very soon show you some pictures. Then we have this snake-like Genopus, and Leucopoxillus is our zombie species because it really strongly grows in a very dense front and even destroyed our chips in the end. This is how psilocybe looks at the hyphal scale. It is growing a little bit giggly, doesn't really seem to know where it wants to go, a little bit back and forth in these chips. And this is the zombie that really takes up all the space it can very quickly. This is still an early stage of the hyphal front coming in. And it grows almost like water would flow, and in the end even destroys our chips. So we have been looking at how these different fungi explore space. This is a chip which was not filled with anything, so there's just air in this. So there's no reward and no chemical cues for the fungi. They grow there through a narrow constriction, which is 200 micrometers long, and then suddenly a pore opens up, and we asked if they explore this pore and try to find some food there, or if they don't care. And we found that there were 2 very clear categories for this character. We had those that always branched and grew around the pore and explored this. And then we had, on the other hand, those that don't care at all. They just always grow completely through. We also included a little barrier because colleagues before have found that an obstacle might induce branching, and we thought, okay, maybe then exploration will be triggered. But it was kind of the same regardless of this little obstacle. Those species that always explored the space did now as well, and those that just grew right through also grew right through, despite this obstacle. We have been looking a little bit in theories of plant ecology as well, and there is this concept of phalanx guerilla dichotomy of space exploration of plants, where a typical phalanx plant really strongly occupies a space and very densely uses the resources, and also very highly competitively tries to exclude other organisms to take over, while a typical guerilla plant is exploring space much more widely, but also much more coarsely, like the strawberry, sending a little new offspring far away to have a new colony, but really disregarding a lot of space in between. So we thought maybe we can use this concept characterizing fungal behavior, fungal space exploration behavior as well. But when we tried to characterize different things we could measure in our chips, we did realize that it was not only that there were some species in between, but most species were in some categories typically phalanx, and in other categories typically guerilla strategists. And we realized that it's actually much better to go for multidimensional space to be able to really characterize this big opportunity of niche separation that the fungi use in soils. Those that have difficulties to grow in certain structures have easy time to grow in a different way, difficult structures, for example. So fungal behavior is much more complex than to put it on one axis only. But going back to difficult structures, we were testing these angle channels and hypothesized that fungal hyphae probably don't like to make a lot of detours, and the more deviating a path is from their natural, most easy, straightforward growth direction, the more difficult it would be for a fungal hyphae to go through. And this is also what we found for most of the species. They were okay with 90 degrees angle in the easiest way of their growth direction. They were less okay with having 90 degrees angles in the square formation. Most did not like at all these very sharp, acute angles, except one, the snake species, actually loved those Z-shaped angles and grew much, much longer in those channels, having found probably a niche that that is not occupied by a lot of other fungi there. Other difficult structures, except those strong acute angles, were, for example, things little corners. This was actually unintentional. We didn't want to make this little corner. It was an error in the design where this channel was moved a little bit too much into the middle of the formal structure. So we have a little corner. And hyphae very often get trapped in there. And they try to push further, but they can't get out and just like coil up behind this this little corner. And also round structures. Fungal hyphae want to grow away from the origin of their culture. They want to radially expand and find new food sources. So they always branch in a way that they grow away from their center. But if we give them a rounded structure, they get fooled and don't realize that we are putting them back towards their origin, and then they happily grow for quite a while back. So this is also a difficult structure for hyphae, apparently. But yes, let's go more into the heart of the question. Can organic matter hide in the soil pore space? And now Micaela wants to take over.
Yes. Hello, everyone. Now we have been seeing how we can use these devices to study fungal behavior, but now we can also use these chips to investigate how organic matter is distributed and how it's degraded by microbes, in this case with the use of fluorescent lab strains and fluorescent substrate. In this study performed by our colleague Carlos, he designed a chip, as you can see here on the left side, that contain different channels with different with different bending angles and different turn orders. And the aim for this is to investigate how the spatial geometry can affect microbial activity, specifically bacterial and fungal growth, their interactions, and organic matter degradation. To do so, we have the chip here, and then there is this reservoir where we fill with liquid nutrient, fluorescent substrate, and it can contain either the single bacterial cells or single fungi, or they can grow together, as you can see on the right side, where red represents fungal hyphae and green are bacterial cells. These chips are then monitored over time with the use of an inverted microscope, in which fluorescent images are taken each day, where we analyze the fluorescence intensity as a proxy for biomass. So how the bacterial cells grew within these channels, where, if you can see here in the graph, we have on the x-axis the different channel types, starting from the smoother ones up to the more complex ones, and in the y-axis we can see the fluorescence intensity. What we can see here, if we see how much they have grown, we can see that bacterial biomass was reduced when the angles were sharper in these channels. And if you can see here in the video, we can see that the bacterial cells are actually acting as robot vacuum cleaners. They are going back and forth from one corner to the other without actually moving farther deep into the chip. And this could have implications of how the organic matter degradation is performed, and also how they can migrate along the chip. As you can see here, when they are growing with fungi, the effect is even stronger. So, of course, the explanation is, for example, that since both of them are occupying the same space, they both are competing for the same resources. And that's why bacterial biomass is reduced. But what about fungi? In fungi, we can see the same as for bacteria, that when the channels have sharper angles, the fungi biomass is reduced. And when the channels are less complex, there is less difficulty for them to move forward, as you can see here in the video. But if they go for these more complex channels, as we saw before for the previous experiment, fungal hyphae have a lot of difficulties to access through these channels. And what it happens is that it actually starts to block the whole access far deeper into the chip. And we can see that, as we mentioned before, when they are growing together with bacteria, fungal biomass is also reduced because of this competition. What about organic matter degradation? Is this affected or not? Well, since fungi, they didn't really consume this substrate, we didn't show the data today. But for bacteria, we saw that the organic matter degradation actually is not influenced by the different types of channels. However, it is influenced when they are growing together with fungi. So as you can see here on the right side, bacteria tend to degrade less organic matter in the very complex channels. And one of these explanations could be that somehow fungi modify the whole environment, making the bacteria less prone to degrade these nutrients. And of course, as we saw before, they are also blocking the access. So that could be another explanation. But if we translate this in a more natural context, we could say that, for example, these more intricate pore spaces make the colonization less efficient for bacteria and fungi. If they cannot colonize these next environments, they cannot really consume the food that is there. And that could be one of the explanations why we can still find a lot of this carbon pool in soil, because maybe they are hidden in these very complex pore spaces. And now that we know that this is happening and how important and determinant is the soil spatial configuration, we wanted to explore this into a more complex level, which is the whole soil community. And to achieve this, we had two approaches. So we used the same chips as they were used for Christine's experiments with fungi. So we buried these chips below ground in a forest for 2 months. And to have a better, a more controlled environment and a better time resolution, we also have them incubated with soil samples at room temperature for the same time. After 2 months of being buried, we collect the chips and we analyze immediately in the microscope. What we saw is that all major microbial groups actually access to these systems, such as bacteria, fungi, and protists. And they not only access and colonize the different spaces, but they also were very well established, same as it would be for natural ecosystems, where we could see like some of the soil food web dynamics, which can help us to understand more of this interaction at the microscale level. As you can see here in this video, we can see how a protist is foraging in a space surrounded by soil particles. In this other scenario, as we have seen before, sharper angles have represent a huge obstacle for different microbes to move in. If you see here, this nematode will have a difficult time to go farther into the chip, and this could be beneficial for the prey which can hide into these narrow spaces. Other things that we could observe is that there are new microhabitat appearing into these relatively large environments for the microbes. And this is because the soil particles start to aggregate, either influenced by the presence of fungal hyphae or by the pore space arrangement, as you can see here in these 2 graphs and in this video, where that you can see here, because of the pore space arrangement, it could retain water films and also soil particles, providing a temporary microhabitat for different microbes, which can be very relevant considering certain drastic conditions or non-saturated conditions in soil. But also what we found and we very, very fascinating and interesting is that fungi, they are key, they play a key role in soil dynamics. And basically they are acting as micro-engineers in the soils, because they influence the distribution of particles, but they can also start to open different spaces, separating other habitats and interacting with different microbes. As you can see here in the video, along the hyphae, soil particles start to bind, and then this creates a completely barrier between this environment that now are becoming 2, which can of course influence the dynamics in each one. But as you remember before, we also had these chips incubated in the lab, which can help us to understand how this dynamic occurs over time. And what we can see here in this GIF is fascinating, because we can see that immediately I have ice growing into these wide open spaces, but then after only 10 days, it started to degrade, providing new resources for other microbes. So the turnover can be super fast. And they also, fungi, they not only provide resources, but they can also help and facilitate microbial dispersal and growth by connecting the pore space with water films, as you can see here, and which has been actually seen in laboratory settings, but with a specific fungal species and also a specific bacterial species. And now we actually see that this can be also seen in soils, and maybe it's more common than we thought. For example, here when we analyzed in these wide open spaces, we saw that when fungal hyphae, they grow into these spaces, they drag water that help the bacteria to access to other pristine environments. And we can see here that the abundance is much higher in presence of the hive. But what was also very fascinating is that fungal hyphae not only facilitate bacterial dispersal, but also facilitate the movement of protists. So when they are colonizing these pristine environments, when there is no other resources, actually they can migrate farther away into the chip. And then when the chips were completely overloaded with water, this facilitation stopped. But it's very fascinating to see how hyphae can help others to colonize and access to other nutrient sources. And now I give the word back to Edith.
Yes, thank you, Micaela. Going back to this allegory of the labyrinth in soils, we actually realized after having worked a lot with these real soil communities, including even soil particles, that maybe our thinking of having a labyrinth like this is not completely correct. It's probably much more like this. And I don't know how many of you know this board game and have been playing this as kids. This is a game board which is partly movable, and each time it is your turn, you take a card, this one here, and you push one lane of these bricks through and push it out on the other hand, on the other side. And this completely remixes the spatial settings and creates new dead ends and creates new connections in this labyrinth, trying to mess up for your friends that you play with. And we think the soil is actually much more like this, because it's really in a very fast, big turnover all the time. And it's also the microbes which are very involved in this. Decomposers are also construction workers. That's maybe easier to grasp thinking of fungi, because they can enmesh things and really like pull things together with the long hyphae that the hyphal mats that they produce. But also bacteria, for example, by producing biofilms, can make very important modifications to the direct surrounding. What we have been talking about earlier is that fungal hyphae can break up very, very strongly new paths in an ecosystem. You probably know roots growing through asphalt, and our hyphae have been growing through rubber material here. They also can clog pores. This is extreme, where an hyphae has been completely filling up a pore by curlingly growing through this. And this little pore nematode is maybe trapped because all the exits of this large spore here have been sealed by a fungal hyphae. We have been studying this a bit in more detail as well by looking at the exudates of fungal hyphae and how they interact with soil components in the direct surrounding. This is fungal hyphae here. It's a microspectroscopy image of the carbon distribution. The blue parts here are exudates. It's the glue, the goo that they exude, and it's wetting this quartz particle, and by this gluing it to its hyphae, and also particles can be glued to each other. A very important first step for soil aggregation. We have been comparing different fungal species again here. This is the vascular mycorrhizal fungus, Rhizophargus irregularis, which had very different exudate chemical composition. And we also have been finding direct chemical modification of the surrounding. Here's a hyphae growing close to goethite particles, which are iron minerals, and we could show that the presence of the hyphal exudates reduced the iron from iron 3 to iron 2, very likely being involved in oxidative organic matter degradation activities. But it's not only hyphae and bacteria that modify their surrounding. Here's a little ciliate, if you can see this fast cell. This is real time. It's really swimming fast like this, pushing through all these soil aggregates or these clumps of soil minerals, and also creating a lot of new channels for other organisms to follow through, which was amazing for us when we saw this the first time, because we had no idea that protists could be either as fast or as strong. That maybe says more about us, but that was an eye-opener for us when we first started. We also have been looking at physical forces impacting soil microhabitats. Here's a chip that we let dry. We had soil on the inoculation entrance that we just let air dry, and then the water in the chip corresponds and also gets sucked out, and we get this almost floodplain-like development of water paths here. And we have been analyzing this with the particle tracking. You can quickly see, I fast forward this a bit, that really areas of fast water movement develop quickly, while there's otherwise areas that have almost no water movement. And imagine if you would be a microbe sitting in this channel here, you would really need to make sure that you're not washed away all the time, but you would also get a permanent supply of nutrients with the flowing soil solution. While if you would be just 30 microns away here, you would experience completely different conditions, very calm, but maybe also not very much food there after a while. A quick outlook what we are recently doing and planning for the future as well. We want to do more chemical analysis in the chip. We have been working with fluorescent markers in on-off trigger modes, but we want to have a higher chemical resolution as well. And this is a lot pushed by Milda, who does spectroscopy, and recently published a perspective about the possibilities to do different spectroscopy techniques, Raman, IR, and also X-ray in chips that need then technical modifications to be able to do this, to be able to measure changes much more in detail. And we're also looking at other ecosystems now and other phenomena and other physical forces. For example, we have some studies ongoing in Arctic ecosystems where soils are exposed to strong freezing thawing cycles that we also study amongst others in the chips. And last but not least, we think that the chips can also be a very important tool for outreach activities, for communicating soil ecosystems to society. And we hope that by being able to see this ecosystem and realizing how alive and how full of organisms it is, that people start hopefully maybe caring a little bit more of the fragility of soil ecosystems as well. There's many other ecosystems that have strong ambassadors fighting for preservation. Maybe we can make a little contribution to make people more aware of soils not being dirt, but also an ecosystem that is worth to preserve. And with this, we want to thank our group and involved people here. And we are very happy to answer some questions.
Great, thank you very much for the presentation. Very, very interesting. Just a reminder to everyone, you can put your questions in the Q&A panel and we'll try to get to as many of them as possible. So I'm very intrigued by the potential impact on soil respiration. Did you see that? I mean, one of the slides you did just a few minutes ago actually showed how the water was breaking it up and breaking up the clumps of the soil, which would, in a way, potentially have increased the percentage of respiration. Did you look at that at all?
We haven't been looking at respiration so far. We are planning to incorporate sensors for oxygen and CO2 in the future, but we haven't done this yet. Those chips were completely water filled, where we were doing the drying experiments until that stage that we showed, at least still. So we would not have CO2 accumulation in there in this setting. We're waterlogged in that state. But we let them dry more, we get also then water-air interfaces, and then these things would also be possible to study. Yes.
Yeah. So how many different parameters do you think you can, from an ideal scenario, can you keep adding? Because, you know, as you look, when we're considering the phytobiome approach, it's really is, can we bring in all these different components? And one of the limitations of many of the technologies is, yes, you can measure 4 or 5, maybe 10, maybe 12, but so what do you see as the potential here?
Our approach is often to single out factors to start with, and to reduce things to really to study the influence of certain very basic factors. Now the first years, but of course you're right, after time we want also to combine factors and study interactions. And yes, we can include more sensors. We can also make the system more realistic. For now it's not very realistic concerning the chemical parts of this labyrinth structure, because it's a silicon-based polymer, not much to do with soil minerals so far, but we can surface treat this polymer. We can change it from hydrophobic to hydrophilic and back. We can even make this partly hydrophilic and partly hydrophobic. We can surface coat it if we want to, or colleagues in the US have been including mineral flakes into the material, mixing it in and exposing them on the surfaces to increase realism. So there's many different possibilities to do in the future.
Have you combined any of the data yet with a microbiome analysis, so to look at what species are actually being impacted in the various channels, for example?
You want to answer this?
Yes. We have not done yet, but we would like to explore this in the future. But to do so, we need to modify the current chips we have now. So this is a plan for the future. So in this case, what we need to do is like our chips, they need that inlet and outlet, so we can create a flow, because now most of the movement is by diffusion. So, and then we can maybe collect all the different organisms that are interacting in a specific channels, for example.
Yeah, I'm making a very beautiful network analysis. In fact, it would be really interesting to see. So, all right, another question has come in. Have you used the system to look at how plant roots impact the fungal and bacterial movements as the root pushes?
We haven't done this voluntarily, though we would love to do this also in the future. We, in a few cases, have gotten accidentally rootlets or root hairs into the chips, but we haven't done this yet in a systematic way. The chips that we constructed are very, very narrow. They have a ceiling of 8 to maximum 12 micrometers. So there's not much space for root in the present construct. We have been doing so because we really wanted to look at single microbial cells, and by this limiting the focal plane for microscopy to single cells. So if we want to include roots, we need to have much larger structures and then go confocal. But this is of course also possible in the future to do.
So are the chips available now, and can they be purchased from you?
No, we haven't started any company for this yet, no. But we're very happy if people mail us, if we can collaborate. For us, producing chips takes, like for a couple of chips, takes 2 days or so. And if somebody wants to reuse chips, there might be possibility if we have manpower availability for this, for interesting studies. It would be very nice to collaborate, of course.
Okay, well, that's good to know, because there may be some people out there who would like to try to build it up and get a much more high throughput production.
Yeah, no company scale yet there.
All right. What are the main gaps or differences between the soil chips simulated in the soil environment for microbial researchers, for microbial scientists?
Pardon, I missed the start.
So the main gaps or differences that you would see between someone who is actually looking at the microbiome, for example, versus what you're doing at the moment? What do you see as potential gaps or even opportunities for exploring?
A difference between what we get into the chips and what we have in the natural soil next to it is that we will very likely have selection for dispersal loving species, because those species that only grow by expanding biofilms will take way longer, and species that, yeah, they don't like to disperse so easily, we will much less likely get into there. We will very quickly, after like 1 to 2 hours, we have usually a chip full with quickly swimming bacteria, and then a lot of other species follow after a while, and the predators and so on. But of course, we will select a little bit for those species that like to disperse and like to explore.
And I would like to add something, because, for example, for the protist community, what we have seen is that when after a certain period, when the chips have been incubated with soil samples, and when the system is very well established, actually we can start to see, because we see a turnover and different species coming and live in the system. And one thing that is very interesting, that after a certain period we can really see very rare species that usually are not seen when we collect soil samples. So that could be also something interesting to look at.
Yeah. So, you know, when you're looking at carbon and you've been focusing on that aspect, how deep are you going for your samples and everything? I mean, because one of the great interests, of course, is what is deep carbon. So, is this a technology that we could use to actually measure that?
We are only starting to use the chips as more monitoring tools. We have been focusing on really basic research questions before, but yes, I mean, we have them colonized by soil. We have been going for the rooting zone so far, but we of course can dig deeper and include them also into the deep parts of the soil. Measure organic matter degradation there, and analyze what organisms we get in there. It's a trap culture kind of. So we have a transparent cyborg-like soil there that gets accepted by at least a lot of the organisms, and that we by this then can take into the lab and analyze, that like voluntarily move into these parts. And yes, we can put this deep down into the soil as well, of course. Do the same things there, but for live monitoring there will also be quite some technical development needed, so we will then for now also dig it out again and analyze it in the lab.
Yeah. So, you know, as you know, in the biologicals industry, they're starting to use a lot of microbial products and putting microbial products into the soil, so having an increased understanding not only of the impact on carbon but on the plant itself and the nutrient, and what's happening to the diversity. Are we increasing the diversity within the soil system? So, you know, these kinds of things could be very useful as we move forward in that arena. Yeah, so I want to thank you again for a really interesting presentation, and we look forward to how you move forward on your next steps, and perhaps we'll hear from you again later this year, maybe at the Phytobiomes Conference, which again, just to remind everyone, abstracts are being accepted now. And thank you for a great presentation, and you already have a potential collaborator who's reached out on the, so you'll be hearing from this particular person who's working in one of my favorite crops, which is wheat. So But I want to thank everyone else for participating today and watching the webinar, and we look forward to all the collaborations that will help us move this entire field forward. So thank you again, Edith and Micaela, and we look forward to seeing you soon.
Thank you very much.
Thank you very much. Yeah.